Now, let's move on to specification number 3, which requires an explanation of enzyme action, specifically referring to the substrate, active site, enzyme substrate complex, and product. As you can see in the diagram here, the enzyme binds to the substrate, forming an enzyme substrate complex and eventually releasing the final product. So far, we've talked about what enzymes are, but now, let's see how these enzymes actually work. Enzymes are made of protein, and their structure is really important. Each enzyme is folded into a very precise three-dimensional shape. This shape is what allows it to do its job properly. Now, within that shape is a special area called the active site. You can think of it as a tiny pocket or space where something specific fits in. That something is called the substrate. It's the substance the enzyme acts on. When the substrate fits into the active site, they come together like puzzle pieces. This forms what we call the enzyme substrate complex. It's basically the moment when the enzyme and substrate are locked together and ready for the reaction to happen. Then, the enzyme helps turn the substrate into a new substance, called the product, and releases it. The enzyme itself stays the same and can be used again. If we break it down step by step, here's how enzymes work. First, we have the enzyme with its active site. This is the spot where a substrate can attach. In the diagram, you can see two differently shaped molecules labeled substrate A and substrate B. These are used to show that only the correctly shaped substrate, like a key fitting into a lock, will fit into the enzyme's active site. Next, the substrates temporarily bind to the enzyme at its active site, fitting in perfectly. This stage is called the enzyme substrate complex. At this point, the enzyme helps join the two substrates together. If you look at the diagram, you can see the enzyme with its active site, where both substrate molecules have attached. Together, they form what's known as the enzyme substrate complex. At the final stage of enzyme action, the substrates have been completely changed, and a new product is formed. This product is then released from the enzyme. As you can see in the diagram, the enzyme is now separated from the newly formed product. The enzyme itself hasn't changed, it stays the same and is ready to carry out the reaction again with another substrate. So, here's what we know now. Enzyme-controlled reactions happen when a substrate comes into contact with an enzyme, and that interaction helps trigger a chemical change. Now, let's go over the main parts involved in this process. These are the key components you need to remember. First, we have the substrate, that's the molecule the enzyme acts on. Then there's the enzyme itself. It's a biological catalyst, which means it speeds up the reaction without getting used up. Every enzyme has an active site, which is a specially shaped area where the substrate fits, kind of like a lock and key. When the substrate binds to the active site, they form what we call an enzyme substrate complex. It's just a temporary combination that allows the reaction to take place. After that, the substrate is transformed into a new product. Once the product is released, the enzyme stays exactly the same and is ready to start the process all over again with a new substrate. For example, the enzyme sucrase acts on the sugar sucrose. After the reaction, the products formed are glucose and fructose, two simpler sugars.